Hi, good afternoon, everyone. There's a lot of you. There's a lot of you. Standing on stage, you really get a sense of the scope. So I'm just going to dive right into it. So good afternoon, everyone. My name is Noah Bryan, and I'm a 16-year-old grade 12 student from Toronto, Canada. And today I'll be giving a brief overview of a platform that I've developed to determine the safety of water for drinking. I'll begin by setting some context for my project and introducing the challenge that I'm responding to. And then we'll wrap up with the description of my methods, a very special highlight of the MinION's role in my test, and finally, some future directions. Now, before I begin, I'd like to ask you all a relatively simple question. How would you define clean water? The truth is that it often depends on the context. Take Lake Wilcox, for instance, a recreational hotspot in my community where people swim, boat, and fish throughout our summer months. My friends and I were heading down to the beach one day when I noticed a sign that indicated the water might be unsafe for swimming. That uncertainty struck a chord with me. A few experiments and publications later, I was able to conclude that Lake Wilcox was not safe for swimming due to bacterial contamination. Several swimmers, in fact, including some of my friends, developed gastrointestinal illnesses upon accidentally ingesting the contaminated water while swimming. This got me thinking. If recreational water can make people this sick, imagine the threat posed by intentionally drinking water from such sources. This is the unfortunate reality for many worldwide. Over 250,000 children under the age of 5 die each year due to diseases derived from inadequate water, which is why UNICEF challenged the world to design a test that could be used to simply and accurately identify fecal contamination in drinking water within 10 hours. My test is unique in that it can detect all the bacteria that are present in a water source within 9 hours right from the water side. First, I use this device that I built to concentrate the bacteria directly from the water source, capturing them in a filter. Using the Bento box, I then purify and isolate the DNA from the bacterial cells, then use the machine's thermocycler to amplify the 16S rRNA gene via PCR. I chose this gene as a target because it's sort of like a unique fingerprint for each bacterial species. I then finally use the MinION, which is a handheld DNA sequencer, as you know, to sequence the 16S genes from my sample and compare them to a database to identify the bacteria that are present. Now, the MinION is the perfect tool for this test for a couple of reasons. Oxford Nanopore software allows me to see the sequencing happen in real time and also gauge the number of new reads at any point during the run. When I see the curve sort of level out, it tells me that I've likely captured all there is to find in my sample, which I find takes about 2 hours. My vision? I see this test being used routinely in rural African village all across, the continent. With the support of Bento Lab's Oxford Nanopore and prospectively the World Bank, I plan to launch a cross-continental training program to train locals to use the test themselves. Due to the quickness and wide coverage of the test, a village could determine which of 2 water sources is safer to drink from within 24 hours, based on the specific pathogens which are present in each. In these communities, the difference between life and death can come down to choosing between the lesser of 2 evils, or in other words, the safer of 2 contaminated water sources. With this test, we can arm these people with the most powerful tool of all in the modern age, information, and truly give them a fighting chance. Ladies and gentlemen, I'm here to convince you that clean water isn't just a dream. It's a future I'm building, one test at a time.